Next week is lab practical on everything since the last time. So muscular, nervous, sensory, endocrine, and reproductive are going to be on the lab practical next week. And after we're done with the lab practical, we do the review session for the exam 4. The week after that, we have exam 4, and we have whatever is the next in the lab. So we are not going. I hate doing the practical and the lecture exam at the same time. We had to do it because of the power outage and to make sure that the two sections are kind of caught up with each other. I hate it for you. I hate it for me. I had to double the grade, and everybody's nervous, and it's much better to have only one exam at a time. All right. So. You got that started?
Yeah.
Very precise.
Yes. Okay.
You go. Lovely. You have all your phones sitting right there on the male reproductive system. I'm not saying anything more. Okay. Reproductive system. All these other systems that we are talking about are systems that are involved in our survival. But no matter how well those systems work, in the end, entropy takes over and we all die. The only way a species can continue is if there's a next generation, a next one, a next one, a next one. So for that, instead of us as individuals remaining alive forever, instead we're going to reproduce, and then our kids and grandkids and great-grandkids are going to continue our genetics and our our genetic background. Okay. Anything specific?
Well, no, I'm going to try.
Okay, to be or not to be. All right. So, so apart from trying to survive as long as possible, the idea, and in many other species, is to try to survive until you reproduce. And there are a lot of species that die as soon as they're done reproduction. You all probably know about salmon, right? They swim up into the rivers and often up the waterfalls and all that stuff in order to go up into the river. They spawn and immediately die, right? And then their eggs hatch and swim back into the ocean. There's a mammal, marsupial, in Australia, where when it's mating season, they stop eating and they just get in a crazy mating frenzy. And as soon as they're done, they just mate for like 2 or 3 days, do nothing else, no sleep, no eating, no nothing, and completely exhaust themselves to the point that they die. The males die, the females then carry the babies. So it's an extremely powerful instinct. It's an extremely powerful drive. It's made to be a powerful drive because of the extremely high levels of feeling of pleasure that the sensory, nervous, and endocrine systems together are providing. It's more fun than food. So that is a very, very strong drive and a strong instinct. So we're going to talk a little bit about how it all works. And we have some models here which we can use. This is a model, and this is also a model, just different kind of way it's projected, of the female reproductive system. Okay, so this whole thing here is the uterus, and then you see this kind of border here, that's the cervix, and then this part here is vagina. Outside part, which is not on this model, is the vulva, all the labia and all that stuff. Here, on each side, are the two oviducts, or fallopian tubes. And then on each side here are the ovaries. Now this model has all sorts of cancers on it and whatnot. We're going to ignore that. Vagina is very much open to the outside world, which means it's very susceptible to infections and all sorts of other things. So it has a lot of very strong defenses, which you'll see it's important in a minute. So it has high acidity, it has some antimicrobial secretions, and has antibodies like IgAs in there, all trying to prevent infection, fighting whatever foreign bodies might come in there. The uterus is where, inside of the uterus is where the embryo and fetus is going to grow, right? And it's wonderfully protected in there. But really, you should think about the uterus in the opposite way. It's protecting the mother from the baby, because fetus has its own interests. Fetus is only half genetically mom and half is dad. Dad has interests too. And so in the interest of the fetus is to grow as fast and as much as possible. And in the interest of mom is to live, which means not to allow these parasites to drink everything, right? And eat everything. And so this uterus is the protective wall between mom and fetus, where mom can control how much nutrition the fetus can get. And there's some issues about mom's immune system versus the kid's immune system, so there's some control there. All of this is happening in placental mammals, right? Most of the mammals are placental mammals, but if you've taken classes in like 112, you've heard of monotremes and marsupials. What are monotremes? Platypus and echidna, those are mammals that lay eggs. No big deal, they don't have to have a uterus, right? They have something like a uterus, it's just a canal for laying eggs. Marsupials are kangaroos and koalas and wombats and possums, and so there's fertilization, there's an early embryo, and then that uterus is small and it's not sufficient to nurture the embryo, so the embryo gets born very, very early when it's really, really tiny and crawls up into a pouch where it latches onto a nipple and sucks milk, and that's how it gets both the immune response and the nutrition from the mother as it grows. Most of the mammals are placental mammals. Where does the placenta come from is a very interesting story, because at some point, some hundreds of millions of years ago, some primitive marsupial caught a flu, or a viral disease of some sort. And you know how viruses work. They get into the cell and get embedded into the genome and hijack the cellular machinery to make more copies of them. So, the viral genome is going to become just like one of the many genes in the genome of the host, and it gets transcribed and translated all the time and makes a lot of new viruses. So, with COVID, there's going to be your various epithelia, respiratory and reproductive and digestive, all sorts of epithelia are going to be full of that virus. In this case, the virus managed to enter the cells of the gonads and the future gametes. What are gonads? Testes in the male, ovary in the female, and they're producing gametes, which are the cells, the sperm cells and the egg cells. So this virus got incorporated either into a sperm cell or to an egg cell that became progeny, became offspring of that individual. And that gene that got stuck in the genome of that species for a while, and probably got silenced after a while, became the part of the junk DNA. And then at some point, as it was mutating, it got restarted again, but now it the body could use it. What it did is altered a little bit the earliest embryonic development of the baby, of that embryo. So when you think about, birds or reptiles or monotremes and marsupials, you have an egg gets fertilized by a sperm, which triggers the first cell division into 2, and then 4, 8, 16, whatnot, many cells, right? And that is going to become the new embryo, the new organism. But in this case, some of the cells became the embryo, perfectly normal whole embryo, but some of the cells instead became a different type of tissue that attached the embryo to the inside wall of the uterus, which means that embryo continued developing a little bit longer than it was normal for that species, a few days longer. What does that mean? That means that embryo was protected for a longer time. The safest place to be is inside of your mom. That big ferocious mama, right? Not everything attacks a big, giant kangaroo, right? So if you're a big ferocious mama with some experience about surviving, then the baby inside of you is pretty safe. Anything else, little crawling thing out into the pouch, maybe latching onto the nipple, maybe not, is not as safe. Egg, of course, can be eaten by a snake or something. So the longer you stay, the better. And so there was suddenly selection for this organ, which is placenta. And so the selection for it is to be able to keep the baby inside longer and longer and longer, which meant that the placenta became more and more elaborate and controlling the nutrition and all that stuff. So if you study comparative anatomy and physiology of various mammals, you're going to learn that different types of placenta, some are very tight, some are very loose and whatnot, but all of them have resulted from that virus. All the placental mammals are offspring, or are developed or evolved out of that one species, out of that one individual who got sick from that virus. Those viruses are called HERVs, H-E-R-V. That stands for human. Oh my God, Okay, I just forgot it. H-E-R-V. V is for virus, retrovirus is RV. Endogenous, that's right. Human endogenous retrovirus. And other species you would call it something different. So SERVs for canine, ERV. And that's how we got a placenta, and obviously very important part of the reproduction. Okay, so here's a much bigger model of the ovary. So ovary is the gonad, and inside of it is going to be development of the gametes, which are egg cells. You have stem cells, which are oogonia, which are going to develop into primary, secondary, tertiary oocytes. And for a long time, for decades, all the textbooks were saying that all of that, meiosis 1 that is producing primary oocytes is happening before birth, and then it stops, and nothing happens until you hit puberty. Once you hit puberty, a few eggs every month get recruited to develop into secondary, tertiary, and then one of them ovulates. And if you're a mouse, maybe a dozen of them ovulate. But if you're human, it's usually going to be only one. Now we know, and that's pretty recent research over the last couple of years, that there are at least some women, maybe it's in various families or whatnot, who are capable of producing new egg cells in adulthood. So what do textbooks do? They don't explain all of that because now it's controversial and there's a lot of research ongoing. They just deleted that statement. One day maybe they'll bring it back, right? Because textbooks have to give you the basics that everybody agrees on, right? you don't do an ongoing new research you don't put that in textbooks, right? So it is deleted out of your textbook. So you should know that at least in some women there's a possibility of making new egg cells in adulthood after puberty. Anyway, so they get, you have oogonia turning into primary oocytes, so they finish the meiosis 1 and they start meiosis 2, and then it stops. And they're sitting in that kind of arrested state for a long, long time. And then every, it says 28 days, it varies between women, it's not exactly 28 days. Every 28 days there's various hormones are going to trigger a few of those primary oocytes to become secondary and to become tertiary. It's not happening in isolation, it's happening inside of follicles. So primordial follicles become primary follicles, become secondary follicles, become tertiary follicles, also called Graafian follicles, which you can look on the slides. And those follicles are maturing, so the egg is maturing while at the same time the follicle around is maturing, getting bigger, getting filled with liquid. There is a layer of kind of carbohydrates and proteins and peptides that is surrounding the egg inside of the follicle. The follicle is built of theca and granulosa cells, both of which secrete estrogen. And at some point, you're going to have several tertiary, so completely mature follicles moving all the way to the surface. And then there has to be a decision which one of those is going to be ovulated. And so there's a competition between them, and there's a multiple set of factors that decide which one will ovulate. And that is the size, the biggest one, the number of theca cells and granulosa cells, and the number of FSH receptors on those cells. So if you have all those three, that's the follicle that is going to ovulate. The others essentially shrink and get reabsorbed. They never get ovulated. And so those theca and granulosa cells are secreting estrogen. Bigger the follicle, more estrogen. More theca and granulosa cells, more estrogen, right? And so for a long time, the whole cycle, more estrogen, more, negative feedback loop. It's going to tell the GnRH to shut down. It's going to tell LH and FSH to shut down. You have this kind of regular negative feedback loop. But at some point, the levels of estrogen are going to get so high that it's going to flip. There's going to be a switch in the hypothalamus that is going to switch it from negative feedback loop to positive feedback loop. Suddenly, more estrogen you have, more GnRH you have, more LH you have, more FSH you have. It's a positive feedback loop. It potentiates. Suddenly you have a huge spike of LH, FSH, and estrogen, right? And that LH spike is going to result in ovulation of that one egg. So the follicle is going to be under the pressure of the liquid. It's producing, those theca and granulosa cells are producing liquid and pumping more and more liquid until the pressure is so high that the follicle bursts. And it bursts under pressure. And under pressure, that egg, surrounded by that layer of peptides and proteins and carbohydrates called zona pellucida, the whole mass is going to be ejected out. Now this thing here, that's the oviduct or fallopian tube. It's not attached to the ovary. It's kind of scanning the ovarian surface, and in hope of catching that ovulated egg. So the ovulated egg goes in there. Extremely high levels of estrogen are also going to stimulate that oviduct to move around. These are fimbria. They're also going to work more. Inside of that are villi, which are going to start beating and making a stream of liquid, which is going to pull up that egg if it gets ovulated and start moving it up the oviduct. about halfway down the oviduct, if there's sperm, that's where the fertilization is going to happen. Fertilization happens in the oviduct. And then after fertilization, the egg already starts dividing into 2, 8, 16, gradually moves, migrates into the uterus, where it embeds into the wall of the uterus. In the meantime, that ovulated follicle transforms itself into what we call the yellow body, or corpus luteum. And instead of secreting estrogen, now it starts secreting progesterone. What does progesterone do is protects the wall of the uterus. At this point of the cycle, the wall of the uterus is thick, has lots of arteries and veins, and progesterone is keeping it that way. It's not allowing it to shed in a menstrual, bleeding. It keeps it thick and full of, the arteries and veins there. And so, if there is a pregnancy and that fertilized egg embeds itself into the wall of the uterus, that uterus is going to send a signal, a chemical signal, a kind of prostaglandin, that tells the yellow body to keep doing what it's doing, to keep alive and to keep pumping progesterone and to keep protecting the uterus, essentially protecting the pregnancy for the next 9 months. If there's no fertilization, there's no signal from the uterus, and the yellow body stops after a few days, does not produce progesterone anymore, and starts shrinking, and it becomes white body, which is called albicans. And it takes a few weeks for it to be completely reabsorbed by the rest of the tissue of the ovary. There's proteins and sugars there and fats that you can use to make other cells and to repair itself. So there's that kind of a cycle. So there's ovarian cycle, the cycle of the egg cell, the cycle of the follicle, and the menstrual cycle. Those 3 cycles are related to each other. And there's a wonderful graph in your book that shows each one of them and what are the levels of hormones in the blood at the same time. I can ask a question about high LH, high FSH, high this, high that, low this. This is what you see in the ovary, this is what you see in the uterus, this is what you see in the oviduct. What day roughly of the menstrual cycle are we in? And your choices are, 0, 7, 14, 21, 28. And you'll tell me, right? So is it just before ovulation, or is it halfway between two ovulations or whatever? So that's what it happens there. So occasionally, the egg gets ovulated, but the oviduct is not on top of it. And it escapes out. Most of the time, nothing then happens. It just, it's a tiny little egg. It just gets dissolved. But sometimes there's sperm coming out too. And there's egg coming out here and sperm coming out here, and fertilization happens outside of the oviduct. This is called what? Ectopic pregnancy. Extremely dangerous, because now you don't have the protection of the uterus. The little embryo can now parasitize on you fully, and you can't protect yourself for it. And it's going to find something that has a lot of nutrients, like a liver or a kidney or mesentery or intestine, right? And latch itself on it and start sucking the life out of you, and you can die. You will die. So that it has to be removed. Which is really hard in some of the states in the United States right now with all these new anti-abortion laws that are covering everything, right? Really, really bad idea, right? Because people will die. And so ectopic pregnancy is a very, very dangerous thing. Okay, so that's about what I'm going to say about the female. Your textbook is excellent, so please use it as a resource for the next exam, not just what I'm saying right now, or the how do you call it, the study guide. All right, what's this? Male reproductive system. Okay, So let's start with the testes. This is the testes. There are two, one left and right. And inside of the testes are lots and lots of tubes. Those are convoluted tubules because, well, they're convoluted. And so the wall of each one of those tubes is going to contain different types of cells. It's going to contain spermatogonia. Those are the stem cells for the sperm, right? And they're going to have cells called Sertoli cells. Those are feeder cells. Those are nutritional and stimulatory cells that are telling and helping the spermatogonia develop first into spermatids and then into spermatozoids. So maturing as sperm cells, undergoing meiosis 1 and meiosis 2. They go all the way. With the egg cell, it doesn't go all the way until fertilization. Fertilization is needed for the meiosis 2 to end. Sperm cells go all the way. Also, when you have meiosis in the egg cells, you have egg cell undergoing meiosis 1, which is going to separate it into 2 cells, and then meiosis 2, that's 4 cells. But you don't get 4 cells because on the way you want the egg cell to have as much cytoplasm as possible. So when it divides only the genome into an egg cell and a polar body, which is essentially chromosomes surrounded by membrane. That's all. So one is a humongous cell with all of the cytoplasm from both, and the other one is just the way to discard extra genome because you don't want it. You don't want a diploid, you want a haploid gamete, right? And the meiosis 2, you get 1 egg cell and 3 polar bodies. So you totally get 1 egg cell and 4 polar bodies from 2 meiosis. In sperm cells, doesn't matter, they're going to be tiny anyway, right? So through meiosis 1 and meiosis 2, you end up, from 1 cell, you end up with 4 sperm cells. As they're maturing, they're moving around. They're moving through the convoluted tubules, and they're going to end up in these straight tubules that are called retatestes, and then they're going to enter the epididymis, first in the head here and then in the tail. And they're going to sit there for a long, long time, and they're actively inhibited from swimming, right? So they're completely mature. How does a mature sperm look like? There's a cell body, which is the head, which is essentially just nucleus, a little bit of cytoplasm around it. It is covered with another layer of peptides and carbohydrates, which is called acrosome, which is necessary for fertilization. And then behind it, there's a neck, which is full of mitochondria, and then machinery for flitting around a very long flagellum, right? The tail that is going to be doing the swimming. So they're really tiny, but when they're sitting in the epididymis, they are actively inhibited from swimming. If there's a lot of them, if they're not ejected, they're not ejaculated, they're going to go all the way into the vas deferens, which is the next tube, which is this one here, and kind of start filling it, a quarter of it, a third of it, or half of it. And at some point, there's going to be a release. Maybe you're asleep, or maybe you're actively working on that, and there's going to be ejaculation. So during ejaculation, all of these sperm cells from the epididymis or whatever may have accumulated in the vas deferens are going to surge very quickly through the vas deferens into the urethra here, and urethra in this part here is going through the prostate, and then it's going through the penis and out into the vagina. This is a very fast process. During that process, a lot of stuff has to happen. There's a bunch of dry cells. And what comes out is not a bunch of dry cells, it's semen, right? So there's liquid. That liquid is coming from 3 different sources. And we're going to go through these sources right now. You see these 2 yellow kind of organs here? Those are called bulbourethral glands or Cowper's glands. And they produce salts. Now, salts act as soaps or detergents. And so, during sexual stimulation, bulbourethral glands are going to secrete their secretions, not a lot of it, a little bit, and it's going to go through the penile urethra, and it's got salts and detergents. It's going to clean it up. Who knows what's in there? A little bit of urine, maybe dust and dirt and bacteria, maybe sperm from yesterday, all sorts of stuff you don't want to put into the woman, right? Into the vagina. So this is pre-ejaculate, right? And it's going to kind of flush it, clean it, so when the new sperm comes through, it's nice and clean sperm and sterile and no other gunk and goo. The prostate is going to add its secretions to the semen, and that is mostly protein. That protein, we're going to talk about it in a second. But the first thing that enters the vas deferens just before it gets into the urethra are the seminal vesicles. There's the one and there's another one. Seminal vesicles are producing most of the semen, or the liquids, about 60%. And they have all sorts of components, but the major component is fructose. All right, so the kind of all these dry sperm cells, which have been inhibited from swimming, are going quickly through all of this vas deferens into the urethra and out, while gaining a lot of liquid from the seminal vesicle and from the prostate. And then it gets into the vagina. And vagina is, as we said before, an extremely hostile place, inhospitable place, right? Vagina has to defend itself from foreign stuff, right? Because it's very easy for bacteria and dirt and stuff like that to get out, because it's a kind of a open space, right? It's a big opening there, and it's very easy for stuff to come in. And so you bring in sperm in there, and they are foreign bodies, and they're going to get attacked by the vagina, right? Extreme acid, antibacterials, and antibodies. So what are the poor sperm to do? They're going to die. Well, they don't die because, remember the protein from the prostate? It coagulates under low pH. So it's very acidic in there. As soon as it gets into the vagina, all of that protein coagulates. Look like, when you cook the egg white. It's like cheese, right? And so all those sperm cells are embedded in that cheese, protecting itself from the attacks by the vagina. So the acidic secretions and antibacterial secretions and antibodies can't reach the sperm cells to kill them. And it takes a few minutes. During those few minutes, what is happening is that the fructose that is coming from the seminal gland is entering the sperm cells and undergoes first glycolysis. It gets broken down exactly like glucose. Those are very, very closely related molecules. And it can be used completely like it gets broken down by glycolysis to pyruvate, which then enters the mitochondria. Remember, there's gazillions of mitochondria in the neck of the sperm. So they immediately start producing ATP, which powers the engine for the flagellum. And now they start swimming, right? So they're starting to beat their tails, and after a few minutes, that coagulum liquefies again. So that whole liquid with the sperm swimming is swimming through that liquid, through the vagina and through the cervix, into the uterus, and all the way to the oviduct. How do they know which way to swim? They're on the tips of their heads, they have 7 transmembrane G protein mediated receptors, which are olfactory receptors. Those are smell receptors. They can smell which way is the egg. And they start swimming that way. Most of them are not going to make it, but a few of them will. And if there's an egg there, a bunch of those sperm cells, the survivors, 24 of them out of billions that were ejaculated, they're going to attach themselves, and they have, remember, the acrosome. a layer of peptides and carbohydrates. At the same time, the egg has zona pellucida, a layer of proteins and peptides and carbohydrates. And at some point, one of the sperm is going to enter the cell. The flagella might stay outside, depends on the species. But one sperm cell enters the cell. That moment, zona pellucida completely changes its consistency, and then no other sperm cell can get in anymore. They're all blocked. So it's very rare to get two simultaneous and then you get twins. And it's very rare that it's happening simultaneously in the left and the right ovary. So again, you get twins. But the moment one enters, zona pellucida prevents any other sperm from entering. And then you have the embryonic development starting. There's whole thing. Now you have a diploid cell again, and you don't need to worry about it. It's going to continue with the development. Okay, this is one of the testes, but it's inside of the scrotum. So the testes in most mammals are not inside of the body cavity. They are outside in the scrotum. Does anybody know why? Why are they out? Because inside is too warm. Spermatogenesis requires lower temperature than the core body temperature, right? So it's really out there to be cooled, and so it's nicely in the scrotum, which is kind of a dangerous place to be, all right? And so it has two layers here of muscles. Those are called dartos muscle and cremaster muscle. Dartos and cremaster muscles have slightly different functions. So cremaster muscle is going to shrivel up the skin of the scrotum, that's increasing the surface area, right? So more of the heat can be lost. While dartos muscles are actually pulling the testes and the scrotum up into the body cavity. So when is this going to happen? It's going to happen when those two muscles are going to stimulate it in several different situations. One is when you're too cold. So when you're running out in snow naked, some people I guess do that, or you're swimming in cold water, right? Taking a cold shower. So when it's cold, the dartos and cremaster muscles are going to be stimulated, and they're going to pull the scrotum and the testes up into the body cavity. Another one is stress response. Remember, you're a zebra and you're running away from the lion. It's not a good idea to have the scrotum, flinging around, right? You want it out of the way so you can run as efficiently as you can. You also don't want it to be injured. You don't want it to be bitten by a lion right there, right? So you're hiding it away, putting it away out of the movement, out of the motion of the legs. So stress is going to do it. And in general, movement, exercise, any kind of movement. So let's say you get in a fight, fist fight with somebody, right? So first you're stressed, so there's going to be a stimulus. And second, you're moving, right? You're kicking and biting and whatnot. And so you and the other person is kicking you, might kick you right there, right? So you protect it by getting the testes closer into the body. So stress, activity, and cold. Coitus is activity. So it's usually going to happen. So sexual stimulation is also going to stimulate the dartos and cremaster muscles to contract, bringing the testes closer to the body. Okay, so good quiz question or an exam question is how do I test you? How do you know what those three types of glands are doing and what are they producing? So this is how I can, I'm going to ask it in different ways, but think of it this way, because you'll never forget after this. So I'm going to give you the secretion of the seminal gland, of the prostate, and the bulbourethral gland. And, this is like 60, 30, 10%. No, I'm going to give you exactly the same volumes in 3 glass jars. And you're going to tell me which one is which without being able to use any equipment, any instrument, anything. How are you going to figure out which is which? There's 3 clear jars, and I gave it in exactly the same quantity, the same volume, so you can't judge by volume. Okay, we're getting there somewhere. Color. Are they going to be different by color or the way they just look?
Like one is going to be more thick than the other one, maybe? One is going to be more thick than the other one?
The prostate one is made of protein, so it's going to be milky, right? The other 2 are clear. And this one is milky. You will say, okay, this is clear, this was clear, this is essentially looks like milk. That's prostate. Okay, how do you differentiate between the 2 others?
Viscosity? Viscosity?
They're both very, very low viscosity. They're essentially water.
There's actual sperm.
There's not going to be any sperm in there. Once you have eliminated prostate because it looks differently, you have still 2 jars of clear, highly low viscosity liquids, and you have to tell me which one is the seminal gland and which one is bulbourethral gland secretion.
I don't want to say smell.
Huh?
I don't want to say smell, but...
What did you say?
I say smell, because you say the 10% is more like soapy, that one's cleans. So I think they smell is different, but I don't know.
This is going to be salty, this one is going to be sweet. It's fructose. So remember, until recently, we did not have tests for diabetes. So what did the doctors do? Taste the urine. And if it's sweet, you have diabetes. So for, centuries, doctors were doing things like this, using taste and smell and whatnot to diagnose things and to figure out what is what. It's only very recently that we are doing everything with machines. So, this sounds like giggle, ha, 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 ha. This is how it was done for a long, long time. Feeling, touching, hearing, tasting, whatever. You are using all of your senses to figure out what's wrong with the patient. The vets do the same things. I had an old vet walk into our barn, and he just stopped. He walked into the barn and says, there's one horse in here that has a particular disease. How do you know? By smell. He started going around sniffing, and he said, this horse. And took the blood sample, took it to the lab, and said the horse had that. And we treated that horse for that. We had no idea. Horse looked fine, right? So he could smell a difference, something in the sweat or something like that of that horse changed the composition, and he could smell it. Old school veterinarian. So that's how you know. Now you know forever.
That's how we'll find out my dad had diabetes, because I think just go outside, there was a lot of ants around it.
That's exactly right. That's how you do it. Yeah, there are all sorts of ways that you can use nature, like animals and plants and whatnot, to see how they respond to your pee or to whatever, to figure that one out. Okay, so we got that. And then, is there anything else I wanted to say about the reproductive system? Okay, so, I don't have a mammary gland here. So I'm not going to ask it for on the lab practical. It's 2 doors down, and I don't want to interrupt there. Mammary gland. Okay, so there are 2 mammary glands. Of course, males have them, but male mammary glands are never activated. In women, there are 4 hormones that are acting on the mammary glands. Those are estrogen, just the development of it. And then there is progesterone. Progesterone is stimulating the growth of breast during puberty and during pregnancy, right? Somebody here has babies, right? And so boobs get bigger when you're pregnant, right? So that's progesterone. And then prolactin is going to tell the mammary glands to synthesize milk, to make milk, and just store it there. And then oxytocin, you're aware of that, you're familiar with that, is going to eject the milk under the stimulation of, the baby chomping on it. So 4 hormones, the estrogen for just the primary development of the breast and the mammary gland, progesterone, growth of breasts in puberty and during pregnancy, and prolactin stimulates the formation of milk, oxytocin, the ejection of milk. So 4 hormones that you need to know about that. So that's both a reproductive and endocrine question for the exam if you want. So I'm going to definitely ask something like that. The development of the reproductive system. So we have male and female. Okay, so as you all know, in most mammals, almost all mammals, the egg always has the X chromosome, while half of the sperm have X chromosome, half have the Y chromosome instead. And so at the moment of fertilization, it's just a matter of luck. About 50% is going to be the X or the Y chromosome that fertilizes the egg. And so the resulting embryo is going to be either XX, which is chromosomally female, or it's going to be XY, which is chromosomally male. And when it's chromosomally male, so female is the default, right? There's going to be a particular embryonic development, and you end up with a female. If nothing else happens, you get a female. But if it's a Y chromosome, there's going to be a switch, right? You need to now make a male out of this. Remember the organizing effects of hormones when we talked about testosterone having a little blip in the early embryo. Well, this comes from this. On the Y chromosome, there is a gene called SRY. And SRY is a transcription factor. What does it mean? It doesn't do anything directly to the body, but it turns on other genes. So there's a cascade of genes that are turned on, starting with SRY, which exists only on the Y chromosome. There are exceptions when it migrated to X and whatnot. Then you have various different other sexes and genders and whatnot. But in the most people, that SRY is on a Y chromosome, and it starts a cascade of transcription events which is going to turn various genes on, which the first thing they're going to do is they're going to transform the initial ovary into a testis. So ovary becomes testis. The ovarian tissue becomes testicular tissue and immediately starts to secreting testosterone for a couple of days. That testosterone diffuses through the entire embryo and masculinizes all the cells. Suddenly all the cells are showing the testosterone receptor being transcribed and translated. Thus they're responsive to testosterone later in life. And then there's no testosterone until puberty. Once the puberty hits in, all those cells are responsive to testosterone. And so you have the development of primary and secondary sexual characteristics. Or sex characteristic, primary would be the growth of gonads and genitals and all that. Secondary would be growth of facial hair, male pattern baldness, growth of muscles, just the kind of the male body type, right? Deepening of the voice because the larynx grows, so you can start talking really, really deep. Most of you women cannot talk as deep as this, right? Some of you can, but I can't squeak like you can. So Those are the secondary sexual characteristics of the male. Another thing that is happening in the embryonic development is that some structures are going to be transformed, right? All these SRY on all these genes are transforming the development of ovary into development of testes. So ovary becomes testes. And then during that blip of testosterone, some other tissues that are starting to develop as female are going to switch to starting to develop as male. The best example is that the glans clitoris becomes glans penis. And then there are some things that are not transformed, but they are replaced. There are two little tubes inside of the early embryo, and one is called Mullerian and one is called Wolffian tube. And Mullerian tube is normally going to develop into, in default, is going to develop into a female reproductive tract. So the oviducts and the uterus and the vagina are all going to be developed out of the Mullerian tract or Mullerian duct. While at the same time, Wolffian duct is going to be just disappear. It's going to be completely, it's going to just fall apart, and neighboring cells are going to pick up the nutrients. In the case of male, so you have all these genes coming that have been turned on by SRY, and there's also a blip of testosterone. What is going to happen is that the Mullerian duct is going to be inhibited and destroyed, but instead the Wolffian duct is going to start developing. A completely different structure is going to be developing, and it's going to become penile urethra, it's going to become vas deferens, it's going to become parts of epididymis. So the tubing is essentially going to come. So so some of the organs are going to just transform from female to male, while others are going to be replaced. You either have the Mullerian duct developing into the female tubing, or the Wolffian duct developing into the male tubing. And then, all sorts of accidents can happen. You can have hermaphroditism, you can have all sorts of other things. So, and we can talk about how much is that normal or not normal as far as medicine, but everybody's a human, everybody has the same rights. So we don't have to discriminate people because they're not exactly on the extremes of being female or male, which is more gender, but they're intersexes as well. So I think I'm going to stop here. You still have about 15 minutes to look at the slides and take a look at these models. All right, I don't know where the